Right, welcome to our B2 exam revision live stream. Okay, we're set up similar to last time in the fact that if you want to access the PowerPoint that I'm going to be going through, you can get that on our science website, so you can go in and get those. You can email the email address communications@folkestoneacademy.com any questions you may have for the live stream. But you may notice this time that at this point in time we've actually got the comments on the YouTube channel turned on, so you're able to comment live, or should be able to comment live on the video. So if you have any questions and you are logged into YouTube, you can actually write a comment straight on there, and we'll be able to see it and answer those comments live. And if there's any discussion there about the science that's being talked about, that would be great as well. And we've hopefully been able to add in that extra layer of interactivity for you, so you can actually interact with each other, interact with us, and ask your questions that way. The one thing I have to say is if it's not working, or if the comments get a bit silly, we do have the ability to switch off or disable that feature, so we will turn it off if it's going to be a distraction to others who just want to watch the information and get the most out of it, because we're all here to try and learn. I know it's like full on this time now. We've got exam season in full swing. You've had English, you've had maths earlier this week, you've got biology tomorrow, and it doesn't get any easier next week as well before the C2 and P2 exams. So we're really into it, and we all want to get the most out of this in the time we've got. So what I'm going to do is I'm just going to start up my little timer so I know exactly where I am. I'm going to follow the same format that I usually do about half-hour talking. See if we've got any questions. If we've got any questions come in, I'm going to then go through those questions for about 15 minutes, and hopefully there's some good discussion we can have there, and then another half hour, and we'll try to cover as much as we can. I won't be going to every topic in B2. The additional sciences, B2, C2, P2, have a lot of content in them, but I have got a decent amount of time to go through quite a lot, but I want to go into detail on a little bit more than in some of the areas. Some of the areas that I feel are going to be ones that will come up. Either they come up regularly in the exams, or they haven't come up in a little while, and I feel that they're due to come up this year. So we'll give it a go. Okay, starting off then, when we start off the biology, the first thing we need to talk about really is the fundamentals. So B2 starts off talking about cells. Now, we covered a bit of it, or briefly covered a bit of it in B1, so we're going to have a bit more on the two types of cell you need to know. There are actually 4 types of cell you need to know. You've also got yeast and bacterial cells, but they tend to come up a bit less. So we're going to focus on these two. We're going to focus on the animal and the plant cell. In the presentation, if you see it, you've got an animal cell there. This is sort of basic drawing of one, with all the bits you'll need to know in it, and some of them labelled up as well. I'm also going to add some information to my flip chart over here. And we've also got a plant cell, and there's certain things in the plant cell that only come up in a plant cell and are not in an animal cell. For an animal cell, the basic structure is got the outside layer, and it's got darker area in it. All animal cells will have this, and this is kind of the most basic version of an animal cell you can have. In there you've got your nucleus. So you've got a nucleus, and that's really important for containing the DNA, okay, which is the genetic material of the cell, so it contains the DNA, and it also controls the cell. Okay, so that's the important features of, or function, sorry, of the nucleus. Okay, it contains the DNA and it controls the cell. Then you also have the outer layer of the cell, which is the cell membrane. Okay, in an animal cell, you only have a cell membrane, and what the cell membrane does is it controls what goes in and out of the cell. So this controls what goes in and out. And the process by which they move in and out through the cell membrane, and indeed the process of any substance moving in biology, or at least GCSE biology that you need to know for B2, is called diffusion. But we come on to diffusion in a bit. We talk exactly about what that is. Then inside the cell you've got a gel-like substance which is called the cytoplasm. And that's where all the chemical reactions take place. So all the chemical reactions take place in the cytoplasm. Okay. So those are the main features of the cell, of the animal cell. There are a couple that come up occasionally, less frequently, but they do come up still. These ones are, if you had little sort of structures in there, all dotted throughout, every cell's got them, they are mitochondria. And I say mitochondria are mighty, and the reason I say they're mighty is because I remember that energy is produced here, or energy is produced is a bad word, energy is released. So release energy through a process called respiration. Okay. So that's what happens in the mitochondria. Energy is released through the process of respiration. And then also you do have structures in there that are called ribosomes. They're, again, less common, but they are there. Well, they're not less common in terms of the period. They're less common in terms of them being asked about them in GCSE papers, I should say. Ribosomes are where proteins are made. So protein synthesis happens at the ribosome, or in the ribosome, on the ribosome. So protein synthesis is what ribosomes are responsible for. So that's your animal cell. The parts that you really need to know, cytoplasm, nucleus, cell membrane, they're the main parts. And then also the mitochondria, the mighty mitochondria, are what releases energy through the process of respiration, which we will talk about a bit later as well, the types of respiration you can get. And then ribosomes are where proteins are synthesized. Proteins are made inside the cell. And that's really important because for cells to grow, or anything to grow, it needs protein. Because protein makes up most of the substance of cells that they're actually built from. So that's why that's important. These 5 things are also found in plant cells. So all of these 5 pieces are found inside plant cells. Everything you find inside an animal cell, you will find in a plant cell as well. So those 5 things are in common. The difference with a plant cell is generally your plant cells are drawn with a regular shape, okay, and they have a double sort of layer to them, because they have another area. They still have a nucleus in them, but they also have a big area in the middle as well. They have your mighty mitochondria, because they need energy, so they're going to have those. But they do also have different little bits in them, which I'm going to use green because they're actually green in colour, which makes the plant green as a whole. And they also still have ribosomes as well. So just labelling up the parts we already know, we've got the nucleus still, which is always easy to identify. We've got the cell membrane, which is actually the inner layer of the two. It's called the cell membrane, and it controls what goes in and out of the cell. We've got our mitochondria, mighty mitochondria, where energy is released through respiration. We've got our ribosomes. And making up the gel inside it is the cytoplasm. Now, I appreciate that these are lots of difficult words, and I often see students getting some of these words jumbled up, but you've got to be really clear. These are the parts of the cell, and common questions are about this is what do you get in animal cells, what do you get in plant cells, what do you get in both? And they ask you things about that. So identifying the things that are only present in the plant cell is very important. The outer layer, the thick layer that I said was round the outside of the plant cell, is the cell wall. Okay? What do we know about walls? Walls are strong, they're tough, they stand upright. Okay, that's what the cell wall does. It provides support and structure for the cell itself, for the plant cell. People seem to think that cell wall is to keep things out of it. It's not. It gives it support and it gives it structure. So, gives support to the cell. Why is that important? It's because plants need to grow upwards. Why do they need to grow upwards? Because they need to get to light. Okay, so plants need to grow upwards towards light. Whereas animals often have a skeleton of some sort inside them that provides that support. Plants don't. So they need that cell wall to actually give them the support so they can grow up towards the light. Similarly, they have what's called a vacuole in the middle of them. Okay? This is called a permanent vacuole if you want to be specific, but a vacuole is fine. So the vacuole is filled with cell sap. And the reason it's filled with cell sap is again to give the cell support. Okay, so it inflates with cell sap and water and other things, and it sort of blows up and it makes the cell stand up and is hard. The word is actually turgid, which means hard. The cell goes turgid, goes hard, so that the plant can grow upright towards the light. Okay, so that's what the vacuole is for. And then additionally, the last thing we haven't mentioned, but that is vitally important, is these green dots that I've put in there. They are actually called the chloroplasts. And I apologise, it's a bit low, I'm getting quite low down here. Okay, not to be confused with the cytoplasm, chloroplasts are parts of the plant cell which contain chlorophyll, which is a tough one to remember, I know, but contain chlorophyll. And that is the site of photosynthesis. That's where photosynthesis happens. Okay, so photosynthesis takes place in those chloroplasts. So these three, cell wall, which gives support to the cell, the vacuole, which is filled with cell sap and provides more support, and the chloroplasts, which contain chlorophyll and are where photosynthesis happens, those three are only found in plant cells. So use your knowledge there to actually say which cell you've got. If you've got any of these structures in the cell, then you know you're talking about a plant cell. Okay? So that's a really good thing to remember. The other two types of cell you get are bacterial and yeast cell. As I said, I'm not going to go into great detail on those. The only thing I'm going to say about a bacterial cell, in case it comes up, is that bacterial cells, what's special about them, is they don't contain a nucleus. They do not have a nucleus in a bacterial cell. The DNA is just in the cytoplasm. They also have circular bits of DNA, which are called plasmids, which are floating around in the cytoplasm. So those are two special features of bacterial cells. They can also have what's called flagella, but they don't have to have them, which are like tails, which allow the bacterial cells to swim about. Okay? And then yeast cells, they're far less frequently asked about, but in case they are, they're almost in between a plant and animal cell. They have a temporary vacuole, which they can have, not a permanent vacuole like this one. They don't have chloroplasts, they won't have chloroplasts, but they will have, and they do have a nucleus, do have cytoplasm, cell membrane, ribosomes, mitochondria, all that stuff as well. Okay, so yeast cells could come up. I'm not going to go into more detail than that, but they're far less likely to than these two, definitely. Bacterial cells maybe, but yeast cells are less so. I mentioned when I was talking about the cell membrane that substances move in and out of the cell through the cell membrane. Now, looking at how that happens, I said the process was called diffusion. So the process is called diffusion. The movement of any substance that they're talking about in B2 is diffusion. So whenever it's a substance moving, it's diffusion. That could be oxygen, carbon dioxide, water, minerals. If they're ever talking about the movement of a substance, diffusion is how it's moving, as far as you need to know for B2 or B2 exam. And the definition of diffusion, hopefully you know, but in case you didn't, is the movement of a substance from an area of high concentration, which means where there's lots and lots of them, to an area of low concentration, where there's not very many. So it'd be like if we sprayed an aerosol in the room. If I sprayed it at the front of the classroom, you'd probably see the particles come out. There'd be lots of them in this area. It'd be a high concentration. The people at the back wouldn't be able to smell it. Eventually it would spread out and it would fill the room until it reached the back of the room. That's diffusion. It's going to happen in animal cells for oxygen to diffuse into them. Oxygen diffuses in because that's what's needed for respiration, as we will look at when we look at respiration. So oxygen will diffuse into the cell and go to the mitochondria where it's used for respiration. In a plant cell, if you remember photosynthesis, carbon dioxide is needed for photosynthesis to take place. So carbon dioxide is going to diffuse into the plant cell and go into the chloroplasts where it can be used for photosynthesis. Okay, so that's some substances that will diffuse. Also, on a bigger scale, diffusion happens in the lungs. You breathe in oxygen and oxygen will diffuse into your blood. Carbon dioxide will diffuse out of your blood into the air, which you'll then breathe out. And that's how diffusion happens in the lungs. It will also happen in the gut as well, so your intestines, where the glucose from the food you've eaten, the sugar, will diffuse into your bloodstream through your intestine walls, and you can actually absorb the nutrients from your food. In a plant, water will diffuse into the roots, or water can get into the roots through diffusion, or so you can think about, or water can get into the cells through diffusion. So the movement of substances in B2 is always through diffusion. You can speed up diffusion, so you can enable it to go faster by doing a few things. You can increase the concentration gradient. Now that's probably the hardest of the factors to consider. All that means is, if you've got an area where there's lots of particles and an area where there's not very many, the particles will diffuse from where there's lots of them to where there's not very many. And eventually this is going to fall, this is going to go up, and the concentrations will become the same. It will spread out. There's lots of particles here, there's not many here, so the particles spread out till they're evenly spread. The bigger the difference in concentration, the quicker it will diffuse. Okay, so that's one way you can speed up diffusion. Another way is by making the particles move faster, and to do that you can increase the temperature. If I was to heat up this beaker, these red particles would be moving faster, so they would therefore spread out faster. Okay, just because increasing the temperature supplies energy, which makes them move faster. And then last of all, you can increase the surface area of a substance if you want it to diffuse into it. So if I wanted it to diffuse into this plant cell, and I wanted water to diffuse in, I could increase its surface area. And that actually leads to another thing about cells, specialised cells. You need to know like a couple of specialised cells. Specialised cells are cells that only really do one job. A good one in a plant is called a root hair cell. That's a normal plant cell, but it actually has a really long sort of thread, like hair, on it. And that increases the surface area and allows diffusion to happen quicker because you've increased the surface area of the plant cell. Also, another specialised cell that's worth knowing is a sperm cell. Sperm cell, its function is to swim to find the egg and then break through to the egg to fertilise an egg. Because it needs to swim, it has a tail, and that swimming takes a lot of energy. So it needs lots of mighty mitochondria to get that energy, release that energy, so it can actually swim the distance it needs to. So specialised cells, you need to know a little bit about them as well. But speeding up diffusion, we can do that by increasing the concentration gradient, increasing the temperature, or increasing the surface area of the substance that's, where the substance is diffusing across. Okay. In this, I said carbon dioxide wanted to diffuse into a plant cell. So carbon dioxide wants to diffuse in. And the reason it needs to diffuse in is because of one of the most important processes, well, the most important process really on the planet is photosynthesis. You must know the equation for photosynthesis. Okay? You definitely need to know the word equation. You could even be asked the symbol equation. Less common, but you could be asked that. The word equation is carbon dioxide plus water, the two things that plants need, produces glucose and oxygen. That's the word equation. You must remember that. If you were to do the symbol equation, you would have CO2, we all remember that, plus H2O, water, carbon dioxide plus water, makes, and this is the really tricky one, and they wouldn't expect you to remember this, this would be given, glucose, which is C6H12O6, okay, plus oxygen, which is O2, okay, if they were asking for the symbol. That is the symbol equation, but it's not balanced in this case. Okay, to balance it, we only need to remember one number, and that is 666. 6 carbon dioxides combined with 6 waters to give 1 glucose, that stays as 1, and 6 oxygens. It's the only number we need to remember, 666, number of the devil if you like, but 666 is the number that you need to remember for photosynthesis symbol equation if they're going to ask you. You must know the word equation though. Carbon dioxide plus water goes to glucose and oxygen. The whole process, as you probably know, requires light energy. So light needs to be taken in for photosynthesis to happen. And sometimes you see light put above this arrow here. It's not necessarily in the equation, but if you want to have it there, that's where it would go. It doesn't go into the equation because light isn't a chemical itself. Light is the energy that's needed for it. So requires light. That light could be sunlight, and in most cases it is, but it can be any light that's supplied in. Where it takes place is in those chloroplasts, which contain the chemical chlorophyll. Chlorophyll is the green pigment that actually absorbs light. Okay, so the plants will absorb light in their chloroplasts, they'll take in carbon dioxide and water to the chloroplasts, and they'll combine them to make glucose and oxygen, which can be released into the cell. Why do they need that glucose though? Well, they need the glucose that's produced by photosynthesis to do, well, 3 things really. Those 3 things are, the glucose initially can be used for respiration, and respiration is the process where we release energy out of glucose to live, to survive. We need that energy. Okay. So respiration is what glucose is used first. If the plant produces enough glucose that it can respire quite happily, so today in the summer it's nice and warm, lots of light, carbon dioxide is always in the air, the plants can take that in, take in water from the ground, and they can do photosynthesis at quite a fast rate. And when they do, they produce lots and lots of glucose. They might not need all of that glucose currently at this moment. Okay? If they don't need all that glucose at that time, they will store some of it. And they store glucose as starch. So any glucose that they have, they can use for respiration. They can store as starch if they don't need it at that point in time. And when they store it, they can use it later on. Think about a potato. A potato is a food store for a plant. It actually stores it in the roots as starch, and then if it requires that energy later on, say in the winter, when it can't do as much photosynthesis, it can break it down and it can actually use the glucose from it. I said there was 3 things it used the glucose for, and the third one is actually to grow. It uses glucose to grow. And there's 2 ways you can think about it doing that. The first is it actually converts glucose into cellulose. And that's used to create the cell walls. So that cellulose is what the cell walls are made of. And it can actually use that to grow new cells, build new cell walls, so it can actually grow. And then the other thing, when it grows, is it can actually combine glucose, apologies it's a bit low, with nitrates. And nitrates it takes from the ground and it actually turns that into protein. So it combines glucose with nitrates, which it absorbs through its roots, and it actually creates protein, and protein helps it to grow. So that's why plants need glucose. They need it for 3 things: for respiration, to release energy. If they've got enough, they store it as starch. They can also then grow with it, so they can turn glucose into cellulose, and they can combine it with nitrates to make proteins. They can actually store some of the glucose as oil and fats in seeds and nuts as well. That's another thing they could do with it. Now I talked about it being summer at the moment and nice and sunny outside, nice and bright, the right temperature, and that means plants are able to do photosynthesis at a decent rate. They can do it quickly. And why is that important? Well, we can affect the rate, the speed at which photosynthesis happens. And we do that by what's called these limiting factors. Limiting factors mean if any of these are in short supply, photosynthesis won't be able to happen as fast as it should be able to. The three limiting factors you need to know are light, carbon dioxide and temperature. So light, carbon dioxide and temperature. They all slow down or speed up, depending on the availability of these things, photosynthesis happening. Graphs that you will get will look very similar to these. Now these graphs you might have to do two things with. You might have to describe the graph. Now describe, you are just saying what you see happening. And explain, you may have to explain the graph, and to explain it you'll have to talk about the science behind it. Remember, describe is just say what you see on the graph, and explain, you have to go into the science. You need to put a bit more detail into it. Okay? So looking just at this light intensity graph, if we were to describe this, we'd just say what the pattern was. As you increase the light intensity, the rate of photosynthesis increases. At this point, the rate of the photosynthesis, at this point of light intensity, you can read off the graph. If you can use numbers in your description, that's brilliant. The rate of photosynthesis begins to slow its increase. And at this point, the rate of photosynthesis stops increasing or becomes level, or levels out, you can say. So that's describing it. Now, talking about the science behind it, you can actually explain that by saying, as you increase the light intensity, the rate of photosynthesis will increase as plants need light to carry out photosynthesis, or light supplies the energy for photosynthesis. At this point, the graph becomes level because photosynthesis can't increase even when light intensity does, because something else must become a limiting factor. And that usually is carbon dioxide. What that means is, even though you're increasing the amount of light available, the plant's trying to do photosynthesis at a really fast rate, it can't actually do it any faster because it can't get any more CO2 to keep up with the speed it could do photosynthesis. So it can't get faster because carbon dioxide is limiting the speed it can go at. That's very similar for if you're doing carbon dioxide concentration or amount of carbon dioxide. If you increase the amount of carbon dioxide the plant gets, it will be able to do photosynthesis faster, until you get to a point where it can't get any faster, and that's because light has become the limiting factor. It's not getting enough light energy, even though you're supplying loads of carbon dioxide, for it to get any faster. Okay? So that's limiting factors, carbon dioxide, what's going on. And the final one is temperature. So what happens when you increase the temperature? Now, I know my students could tell me that temperature is related to enzymes. So photosynthesis is actually carried out by enzymes, or enzymes are involved in the process. So when you increase the temperature, you're supplying energy in that allows more collisions to happen, and the rate of photosynthesis, the speed of photosynthesis, can become faster until about 40 degrees Celsius. At that point, if you get hotter, the enzymes that control photosynthesis become denatured. Their shape changes. We'll go into enzymes again in a bit, and the rate of photosynthesis slows down, so the rate actually falls. Okay? So that's what's happening with temperature. As you increase temperature, the rate of photosynthesis gets faster, and once you get past about 40 degrees, it starts to slow back down as the enzymes become denatured. Okay? Those are the factors that affect the rate of photosynthesis, or the speed of photosynthesis, how fast it can happen. Farmers, or people who have greenhouses, will be controlling these conditions to try to maximise the rate of photosynthesis. And they occasionally ask you questions about how a farmer could use these factors or use photosynthesis to boost the growth of its plants, or his plants, or her plants for that matter. They could do that by controlling the amount of light the plant gets. How could they do that? Well, in the day, that's fine, they get light from outside, but what about at night? The plants would stop growing at night, or stop being able to do photosynthesis. So you could actually have artificial lighting in your greenhouse, which would supply the light energy needed for the plants to grow. So that's one way you can use this. How else could they do it? Well, there's a certain amount of carbon dioxide in the air normally, but you can actually, if you've got a really high-tech greenhouse, you could pump carbon dioxide in to increase the rate of photosynthesis. They wouldn't pump it up to an incredibly high level, but you could certainly boost it from about 0.03% to about 1 or 2%, and that would be enough to make a big difference to the plants, speeding up the rate of photosynthesis. So you can increase the amount of light by putting artificial lighting up. You can increase the concentration of carbon dioxide by pumping carbon dioxide into the greenhouse. And then, very important in the greenhouse is to control the temperature. So, as I said, the optimum temperature for photosynthesis to occur is about 37 degrees Celsius, same as our body temperature, the temperature that enzymes work best at. You do not want it to get any hotter than that because enzymes will denature and they will stop working. And you don't really want it to be any colder because the enzymes won't work effectively. So you can have heaters in the greenhouse if it's in the winter to keep it at the right temperature, or if it's in the summer you could have remote controlled windows, sort of like we have in the science labs here, which will actually open if it gets too hot to let some of the heat out, so that the plants don't get overheated and then the enzymes stop working, denature, and the plant dies. So that's limiting factors of photosynthesis and how we can use them to good effect. Tied in with cells is levels of organisation. Now what that's talking about is we're all called multicellular organisms. Multicellular, multi means lots and cellular means we're made of cells. So we are multicellular organisms. We are organisms made up of billions of cells. And any multicellular organism is built up in levels, so it has levels of organisation. It starts with a cell. Cells are the building blocks of all life, so we start with our single cell. A group of cells working together to carry out a specific function are called tissues. So if you put cells together all to do the same job, they become a tissue, or you call that a tissue. If you have a group of tissues together which perform a certain role, then you've made an organ. And if you have a group of organs together to do a particular job in the body, you've got an organ system. A few organ systems you should recognise are things like the digestive system, the circulatory system, the heart and blood vessels, maybe the nervous system, the brain and the spine and the nerves that go through the body. These are some organ systems you might recognise. The main one to focus on is the digestive system, I'd say. That's not to say to neglect the others, know a little bit, but know some organs in the digestive system. It is one that is in the spec, so will come up or certainly could come up. With tissues, so going back to tissues, you need to know there are 3 different types of tissues in animals and 3 different types of tissue in plants that you need to recognise. Okay? In animals, the first type of tissue is muscular tissue. Muscular tissue is your muscles. It allows you to move. So muscular tissue is in all animal organisms. Then you've got glandular tissue as another tissue you need to recognise. That releases hormones and enzymes into the body. So glandular tissue. And then this one's actually really tricky. It's called epithelial tissue. And it covers parts of the body. So it covers the organs and covers different parts of the body. So those are the 3 types of tissue you need to recognise in animals. And then also, you've got 3 types of tissue in plants. You've got the mesophyll, or mesophyll. This is where photosynthesis takes place. So most photosynthesis for the plant takes place in that tissue. You've got the transport tissues, and the 2 you need to recognise are the xylem and phloem. The xylem transports water around the body. Okay? And the way I remember that is because my alphabet goes WX, so water is transported by the xylem. Okay? That's what moves it about a plant. So plants have veins, and in the veins are the xylem and the phloem, and water moves through the xylem. And then the other tube you need to remember is called the phloem, the other transport tissue. And that's because food moves in the phloem. Okay? I'm not just really terrible at spelling, it's just a way of remembering that food moves in the phloem, okay? It's just the sound that they make. pH sound gives us an F, and we've got a pH in phloem, so food moves in the phloem. The food for the plant is obviously glucose. That's what we're talking about when we're talking about the food for a plant. And then last of all, you've got the epidermal tissue. That covers the outer layer of the plant. That's the outer layer. Epidermal, the epidermis, is the outer layer. And we can look specifically at the leaf when looking at these tissues. So we need to say how a leaf is adapted to carry out photosynthesis. So how is this leaf adapted to make sure it can do the most photosynthesis possible? This can occasionally be a 6-mark question. It could certainly come up in some form or another, and you need to recognise the different bits to it. So the tissues in here, we've got the epidermal tissue, which is the top and bottom layer that coats the leaf. This is a cross-section of a leaf, if we were to slice through it, a little cross-section. Then in the middle, this whole area is the mesophyll, okay? And there's 2 separate sections to that. There's the palisade layer, which is a row of cells standing up on end. And those contain loads of chloroplasts. And they're right at the top of the leaf, because the top of the leaf is the area that gets the most sunlight, okay? So because the most sunlight is coming into the top of the leaf, this is where you want the most chloroplasts to absorb that sunlight and carry out photosynthesis. Below that you've got the spongy layer. That is like a sponge. You could squeeze it, you could squash it, it's got air gaps in it. And the reason it has air gaps is so that gases can move through it. Carbon dioxide can move into the leaf and diffuse into the cells, and oxygen can move out and diffuse out of the leaf. Okay? So that's why the spongy layer has air spaces in it, to allow that movement of gases. Not shown on this diagram is the transport tissues, but the transport tissues will be in a vein somewhere in there. So if there was a vein in this diagram, that's where the transport tissues would be found. Water going through the xylem, WX, and food going through the phloem. Okay? Then you've got the epidermis, and the lower epidermis has actually little gaps in it called stoma. Those little gaps can open and close to allow gases to enter or stop gases leaving the plant. So they can actually control the passage of gases in and out of the leaf. And the last bit maybe to note is this bit at the top, which is the waxy cuticle, or the wax cuticle, which is a waxy layer. If you ever feel a leaf, the top of it is waxy, and that's actually because wax doesn't mix very well with water, and it actually prevents water from evaporating out the top of the leaf. Why would the leaf want to keep water inside it? Well, because it's needed for photosynthesis. So the way it can prevent water escaping is having that wax cuticle, so water can't actually evaporate out the top. Linked back to photosynthesis and limiting factors, there's actually a practical they can talk about with this. They can talk about the effects of light intensity, or carbon dioxide for that matter, and the effect it has on photosynthesis. And they do that experiment with pondweed. So they'll do a bit of pondweed in a little like funnel with a test tube over the top of it. Now that's my beautifully drawn pondweed. And then they'll put a funnel over the top, like that, and they'll have a test tube over the top of there. And this will all be in a beaker underwater. And what they actually do is they actually put a lamp here with a light bulb in it that provides light energy to the plant. And they can change the amount of light it receives by altering the distance that that lamp is away from the plant. If it moves further away, the light intensity will be lower. If they move it closer, it will increase the light intensity. With that, what they actually do is they count the number of bubbles of gas, oxygen, that is produced by the plant, so by the pondweed. And those bubbles actually go off and float off to the top, and they'll fill up that tube, and it will actually, that's how they measure the rate of photosynthesis, the number of bubbles that are produced. If they are changing the light intensity, they will need to make sure the amount of carbon dioxide in the water stays the same. If they're actually they'll also need to control the temperature, so they might use a heat shield to stop heat from the bowl reaching the pondweed to keep the temperature the same. What they might do, they might try seeing the rate of the effect of carbon dioxide, and they can do that by adding a chemical, sodium carbonate usually, into the water. Then they would need to keep the light intensity the same and the temperature the same as well. Okay? But that's a specific experiment they can do to link to photosynthesis and light intensity. Right, at this point, we don't have any questions, so I'll carry on going through. We're going to move on to respiration, but do remember, if you do have a question, you can email it in to communications@folkestoneacademy.com, or you can actually comment on the video. You can write on there, so if you did have a quick question, that's something that could be done and we can answer that really quickly. But otherwise, we're going to move on and we're going to look at respiration. Now, respiration is the process that every living thing does. And it's not the process of going, okay, that gets confused, but that's just breathing. Okay, respiration takes place in your cells, in the mitochondria. Okay, it's different to breathing. So it's happening in the mitochondria of your cells, and it's controlled by enzymes. There's two versions of it that you actually need to be able to identify and recognise. Okay. The equation for one of them is very easy. As long as you remember the equation photosynthesis, aerobic respiration is easy because it's the opposite. Glucose plus oxygen makes carbon dioxide and water, rather than carbon dioxide and water make glucose and oxygen, as in photosynthesis. So this is exactly the opposite reaction to photosynthesis. Aerobic, what that means is, that means this is happening with oxygen. So oxygen needs to be present. So you have to be able to breathe, take in oxygen, absorb it by diffusion through your lungs into your bloodstream, and that blood has to be pumped around your body and get to the muscles, get to the mitochondria where it's needed for aerobic respiration. If for whatever reason you cannot take in enough oxygen to do aerobic respiration, so that means if you're doing a period of intense exercise and your body cannot take in oxygen fast enough to do aerobic respiration, so you might be sprinting, it will have to do anaerobic respiration. And anaerobic respiration is without oxygen. Aerobic, air with oxygen. Anaerobic, an means without, so without oxygen. The equation for that is simple, it's just glucose goes to lactic acid. And this releases energy as well. It releases a bit less energy than aerobic respiration, but still releases energy. So the body is capable of functioning without oxygen for a short period of time. So if you are sprinting, your muscles are working really fast, they need lots of energy. They can't pump oxygen around the body quick enough to actually do aerobic respiration. So they have to work anaerobically. They can only do that for a short span of time, because this lactic acid actually starts to build up in the muscles, and it's actually poisonous. So you don't want too high levels of this building up. So you actually can only do anaerobic respiration for a short period of time before your muscles become fatigued, they become tired, and they can't actually function. They might start to cramp because there's too much lactic acid built up in them. So your body actually ends up having to break down this lactic acid, and the way it does that is by paying an oxygen debt, it's called. So it means when you've stopped doing anaerobic respiration, you've got lactic acid to break down, you have to take in big deep breaths so that you can take in enough oxygen to do aerobic respiration, but you need to take in extra oxygen so that you can break down the lactic acid that's in your muscles, and it actually gets broken down back into carbon dioxide and water, same things that are produced here. Okay? It's called an oxygen debt. You've got to repay the debt of oxygen that you've got because you were respiring without oxygen for a period of time. With the respiration process, you might need to say about effects of exercise on the body, and that's generally quite common sense, but you need to remember what are the short-term effects and what are the long-term effects of exercise. Some of the short-term effects are, if you're doing exercise, your heart will beat faster and you'll breathe deeper. The reason being, you need to pump more blood around your body to bring the glucose and oxygen to your muscles, and you'll need to breathe deeper to take in more oxygen so that you can do aerobic respiration, which requires oxygen. That's the short-term effects. Some of the longer-term effects of exercise can be building muscle, losing weight, better fitness, cardiovascular fitness. Those can be some of the longer-term effects of exercise. But for the short term, it's increased heart rate and deeper breaths or more rapid breathing can also be an effect. Okay. So that's respiration. It's going on in the mitochondria. It's going on all the time. It's going on inside you now. It's going on inside me. Everybody has to do it, otherwise their cells won't be able to survive. They need that release of energy by respiration inside the mitochondria. It's still controlled by enzymes, so that's why it's important that you regulate your body temperature, because this gives out heat energy as well. So the more respiration you're doing, i.e. when you're doing exercise, your body temperature will increase. And you want to keep that about 37 because enzymes work best at that temperature. You need the energy produced from this, well, to be able to move, to regulate your body temperature. You need that energy, and you also need that energy to grow. So you actually build up proteins by combining amino acids to make proteins to grow. So that actually leads us in quite nicely, because it's a process that's controlled by enzymes, into looking at enzymes. And enzymes tends to be a topic that comes up very regularly, but it's also one we do tend to do quite well on, because we seem to get the understanding of it. The first thing you need to know about enzymes is they're actually proteins. They're long strands of protein which are folded up into a certain shape. And that protein, that long strand of protein, is made up of blocks of amino acids. So enzymes are proteins which are made up of amino acids. The definition of an enzyme, really in its most simple form, is it is a biological catalyst. Okay? It's a catalyst that works in a living system, so a biological catalyst. That means it speeds up a reaction. So speeds up a reaction, makes a reaction happen faster. Okay? All your reactions in your body are controlled by enzymes, pretty much. So they all rely on enzymes to actually function. So they all rely on enzymes for all those processes to happen. And the actual mechanism that they work via is called the lock and key mechanism. And you can show this in an exam. You can always give a diagram if you don't want to describe what's happening. If you asked, explain the lock and key mechanism or show how the lock and key mechanism works, you can do it via diagrams. There is a diagram there, but I always like to draw my enzymes simply as Pac-Man. Okay? And this is the enzyme. You need to label up your enzyme. So this is my enzyme here. And the substance that's going to react with the enzyme, or the enzyme's going to help to react, the reaction's going to catalyse, is called the substrate. And the substrate has a certain shape as well. The reason it has a certain shape is because it fits into this area, which is called the active site. Pac-Man's mouth is the active site. The active site is the certain shape of the enzyme where the substrate will fit into. So that substrate will fit into that active site, and it actually forms what we call an enzyme substrate complex. Okay. In fact, I should label this as well, the substrate. That's just where you've got your enzyme with the shape, and you've got your substrate fitting in there, and they're joined together. That will cause a reaction to happen. So the reaction might be it breaks this substrate up, so it might break it into smaller pieces. And once that's happened, the substrate will actually leave the enzyme or move out. And it will actually return. So it breaks it apart and the enzyme releases it. Okay, and the reaction has happened. That enzyme can then go back round and take another one of the substrates once it's been released. That's called the lock and key mechanism because only one substrate will fit into one active site. Okay, that's what you need to really remember for GCSE. One substrate will fit one active site. So one enzyme catalyses just one reaction. It speeds up that reaction and then it can be used again. So one enzyme can actually catalyse, speed up lots of a reaction. So you need a small amount of the enzyme to catalyse a lot of a reaction. So lots and lots of versions of a reaction. So that's great if everything's going nicely. And as I say, enzymes work best at 37 degrees, which is our body temperature. So enzymes can actually be messed up a little bit. If you increase the temperature above 37 or above 40 really, the enzyme becomes denatured. Okay, the enzyme gets denatured and it actually changes its shape, and that means the shape of the active site actually changes. So it used to be Pac-Man, but now it's sort of melted Pac-Man because he's got too hot, and the active site has changed shape. So the substrate, which used to be able to fit in, can no longer fit into the active site. And we say that the enzyme has become denatured. Okay, you can't say the enzyme has died because enzymes are not alive, they're not living things. They're part of living systems, but they're not alive themselves. So the enzyme doesn't die, and you will lose marks if you say that. You have to say the enzyme has become denatured at a high temperature. Okay, that's what actually happens. So that's enzymes and how they work. Now enzymes are particularly prevalent, come up a lot, or are involved a lot in digestion. And I don't know why, I have a bit of a gut feeling that digestion is going to come up in the exam, but like I say, that's just looking at the different papers that have come up. That this is a topic that hasn't come up in a little while. So there may be a question about it, there may not. I wouldn't base all your revision on it, but if it does, these are the bits you need to know. So in the digestive system, it's got several different organs. As I say, you've got the mouth, or the salivary glands, particularly in the mouth. You've got the esophagus, which is the food pipe. You've got the stomach, which is a ball of muscle designed to churn up food. You've got the pancreas, which releases digestive enzymes or digestive juices into the food. You've got the liver, which produces bile. You've got the small intestine, where food is absorbed or nutrients are absorbed. You've got the large intestine, where water is absorbed, and then the rectum and anus, where food is expelled or waste products are expelled after digestion has happened and the good stuff has been absorbed out of it. So everything in the digestive system is designed to speed up the process of digestion and absorption. Okay, so that's what happens in the digestive system. Sole purpose is to speed up the process of digestion and absorption. Okay? You need to know how that works. You need to know the different bits in it that are specific to breaking down food and absorbing food. The enzymes that are particularly in there are amylase, protease and lipase, and you need to know those and what they do. Amylase is a specific type of carbohydrate. So amylase is a specific type of carbohydrate. Okay? Carbohydrase is an enzyme. You can tell it's an enzyme because it ends in A's. All of these actually end in A's. Protease, lipase, all end in A's. So that's how we can tell they're all enzymes. Amylase is a carbohydrate. What it does is it breaks down starch into sugars. So it breaks down the starch that you eat in the food into sugars. And sugars can be absorbed through the intestines into the bloodstream. And where it actually starts working is works in the mouth. And it's produced by the salivary glands, which are in the mouth. So the salivary glands produce your amylase, which is a type of carbohydrates, because starch is a carbohydrate, and breaks it down into sugar. And it works in the mouth, produced by the salivary glands. Then you've also got protease. Okay. Protease breaks down proteins, and there's no real surprise there because it's called a protease because it works on proteins. And it breaks them down into amino acids. Okay. So it breaks down into amino acids, and where that happens mainly is in the stomach. So that takes place in the stomach. That's the main site for proteases, particularly pepsin, which is a protease they talk about, but they'll give you that name if they're talking about pepsin, works in the stomach. We'll come back to why that's important in a second. And then you have lipase as well. Lipase breaks down lipids, and lipids are the scientific name for fats. So it actually breaks down fats. But we call them lipids. So lipids, fats, are broken down into fatty acid and glycerol. Okay. So that's where that happens, and this happens generally in the small intestine. Okay. And again, we'll go into why that's important. So those are the specific enzymes. Amylase, which is a type of carbohydrates, breaks starch into sugars, starts working in the mouth. The salivary glands are what releases it. You've got protease, which is produced in the stomach and breaks down proteins into amino acids. And lipase, which breaks down fats, lipids into fatty acids and glycerol, and happens in the small intestine. Now, I said the whole of the digestive system was designed to speed that up or speed up that process. And the way it does it is because when this food reaches your stomach, the proteases are released. And what are the conditions in the stomach like? You need to remember that the stomach contains hydrochloric acid. Hydrochloric acid is the specific name of the acid that's in the stomach. And that makes it acidic. And some enzymes, not only does temperature affect them, but pH affects them. pH is how acidic or alkaline a substance is. Pepsin, particularly, which is a type of protease, works best in acidic pHs. So it works in the stomach because there's hydrochloric acid. So the hydrochloric acid does two jobs. It helps to break down food, because it will break down food on its own, and it also provides the correct pH for protease to break down proteins into amino acids. Okay? So two jobs of the stomach acid. The stomach itself is a ball of muscle, lots of muscular tissue, which churns the food up into like a pulp, so it can go through the rest of the digestive system. Then, after it goes out of the stomach, it's still very acidic, it's with lots of stomach acid. It goes past the liver, and the liver, I said, produces a chemical called bile. And bile is a greenish-like fluid which actually neutralises the stomach acid. So bile neutralises stomach acid. You need to know two jobs that bile does. One, it neutralises stomach acid, and the second job it does, and this is the one that often gets forgotten, is it actually emulsifies fats. Now what that means is fat sticks together, so it'd be a big lump of fat moving through your digestive system, and you don't want that. So the bile actually turns it into tiny little droplets, which increase the surface area and make it much easier to break down, make it much easier for the lipase to break it down. So bile does two jobs: neutralises stomach acid and emulsifies fat. That then passes into the small intestine, and when it does, it goes past the pancreas as well, which injects a load of these enzymes into it. It injects your lipase into it. It does also put some more amylase and some different types of proteases into it as well to help digestion. And they work on it when it's in the small intestine. The small intestine, though, is also where food is absorbed. So the small intestine, if you look to the surface of it, would have tiny finger-like projections on it. And those finger-like projections are called villi. and they increase the surface area of the intestine so that food can diffuse into the bloodstream quicker than it would if it was just flat. So villi are little finger-like projections on the surface of the small intestine which increase the surface area so food could be absorbed quicker. It diffuses into the blood quicker in the small intestine. So that's the bits of the digestive system we need to be aware of, and it's also the digestive enzymes, the amylase, protease, the lipase, that we need to be able to recognise as well. Right, we've done about an hour now, so we're well on our way to sort of getting through. We haven't had any more questions come in at this point, so I'm going to carry on again still, but if you do have a question, remember you can ask it. I'm going to move on to cell division, which is a really big topic, and so it'll take a bit of time to go through that, and then if we have some time at the end, I'll go back to some things that I maybe haven't gone into as much detail on throughout. So, cell division. Now, the first thing to know about cell division is inside the cell you have the nucleus, and that's where I said the genetic material was found. The genetic material is the DNA. DNA is like a long strand, a long thin strand, a long piece of string if you will. And when it's coiled up, that DNA is coiled up into a chromosome, it actually coils up and becomes a chromosome. If you were to roll out that DNA, stretch it out, a section of DNA is called a gene. It's actually a gene, a small section of DNA which controls a certain characteristic. And that characteristic is how we inherit different characteristics from our parents. So, there's two ways a cell can divide. There's two types of cell division we need to know a bit about. We need to know the basics. So inside the nucleus you find chromosomes. Chromosomes are coiled bits of DNA, and one part of DNA is a gene. Okay? The two types of cell division are mitosis and, I say, meiosis are the two types you need to know. Mitosis is the one that happens everywhere in our body. It's used for growth and repair, so it makes all the cells in the body. It's used for us growing and repairing our body. So if we cut ourselves and we have a cut, that will heal up and the cells will divide to heal over the cut by mitosis. I sort of remember mitosis by mitosis made my toes. So mitosis made my toes. In mitosis, I'll go through the process. There's several stages to it. The important bits you need to know, apart from the fact it makes all the cells in the body and it's used for growth and repair, are that it actually divides just once. So division happens only once. And then it produces two identical daughter cells to the starting cell. So in mitosis, what it looks like if we were to do it. You actually start with your cell, and there's one bit of important information I did almost miss. Okay, if we're talking about humans, and occasionally they talk about human cells, okay, we're talking about the number of chromosomes in there. A normal human cell has 46 chromosomes in it. And that's actually 23 pairs. Okay, so 46 chromosomes remain 23 pairs. Those pairs are set up because 23 come from your mother and 23 from your father. Okay, so 46 chromosomes in total. I'm not going to draw 46 chromosomes in my cell because it would be too complicated. So I'm only going to draw a couple. So we've got two there, and that's paired up with these two. Okay, so we've got paired up chromosomes. Now, the first thing that happens is it actually duplicates, copies all the chromosomes that are in it. So it will copy the chromosomes. So it'll actually double the number in the cell is the first stage. It will double the number of chromosomes that are present. Make copies of itself. We've got little ones as well. So it doubles the number of chromosomes. So it starts with 46 in a normal cell. So this could be a skin cell, for example, would have 46 chromosomes. The first thing is it actually makes a copy of all of the chromosomes, doubles the number of chromosomes. So doubles the number of chromosomes, or duplicates the number of chromosomes. So it's got twice the amount at this point. And those chromosomes actually line up along the middle. They line up along the centre of the cell. And I could add layers to this, I could add levels to it, but really all you need to know is that cell then pulls apart and divides into two new cells. And those two new cells that are produced are, in every way, identical to the cell that it started with. So it's produced identical copies, clones, of the cell that it started, the parent cell. So this had 46 chromosomes. If we're talking about humans, and these ones here have 46 and 46. Okay, they have the full amount. Right? That's mitosis. Mitosis makes all the cells in your body. It made my toes. It's how my toes grew by mitosis. Okay, 46 chromosomes, doubles the number of chromosomes first of all, then divides only once and produces two identical daughter cells at the end. Okay. Then we've got meiosis. Meiosis makes the gametes or the sex cells. Okay, so meiosis or meiosis makes the gametes, which are the scientific name for the sex cells. I remember this by meiosis made me. Okay, it made the sex cells that made me. It made the sperm and the egg that fused together to make me. So meiosis makes me. The important bits about meiosis is there's actually two divisions. It actually divides twice. And it actually produces 4 different daughter cells through the process. So we're going to talk about a human again. So humans have 46 chromosomes, 23 pairs. So we're going to use that. They could ask you actually about a different organism, but if they did, you wouldn't be expected to know the number of chromosomes. They would tell you, but you need to know that if you start with 46 in mitosis, you end with 2 cells with both with 46. Whereas meiosis is a bit different. So we've got our cell and we've got our chromosomes in there. And the first thing is that it actually duplicates up again. So it duplicates or doubles, copies the number of chromosomes. So we've got twice the amount again. Okay. Then, in meiosis, something that does happen, it's a less talked about thing in GCSE really, but it's still important to the process. It's actually, when they've paired up, it swaps little sections of DNA. So it doubles the number of chromosomes, copies the chromosomes, and then it swaps little sections of DNA. You're not going to be able to see it very well, I admit, on my drawing, because it's probably too small for you, but I would swap little sections, little colours of this these bits of DNA to produce some new ones which are different to the parent ones. But as I say, you're not really going to be able to see that very well, but it doesn't matter. It's the fact that little sections of DNA have been swapped and we've got a different combination in each pairing. Okay, so they've swapped small sections of DNA, and that increases the amount of variation. So, and that leads to increased variation. The cell will then divide, just like before. So it will divide to produce 2 new cells. And those cells will have 46 chromosomes in these cells at this point. So we started up here with 46 chromosomes in our parent cell, and these ones, after the initial division, have 46. But I'm not going to write that up because it's not so important, because it doesn't really stop at that point. Because what happens next is important for meiosis, okay, is that it actually goes on to divide again. Each of those goes on to divide again. Now when they divide again, they haven't duplicated the chromosomes at that point. So they've only kept the same. Now, it's probably not incredibly clear on my diagram, but when they have divided again, because they haven't doubled up the number of chromosomes, the starting number here was 46, 23 pairs. Now, at this stage, each of these cells only has half the number of chromosomes of the parent cell. Only has 23. And that's vitally important for meiosis, to produce the sex cells. These could be the sperm cells in the male, in which case this would have come from the testes, a testicle cell, or they could be egg cells, in which case this would be the ovary, okay? Depending on if it's male or female. And it's important these only have 23, because what's got to happen in fertilisation is that one of these male gametes has got to meet with a female gamete and fuse together, and when they fuse, what's going to happen? If they have half the number, they will fuse to produce a new cell with 46 chromosomes. That cell will go on to divide to become an embryo, and that embryo will contain unspecialised cells, which are called stem cells, which can go on to do any job in the body. Those stem cells can be used to do anything before they've become differentiated or specialised. And when they become differentiated or specialised, they then go on to only really be able to do one job. If they become bone cells, they can only do that. But what could be useful about those stem cells, those initial stem cells, they could be used as a treatment. Say someone's damaged their spine and their spine won't repair, you could actually use stem cells to fix the damage in their spine, and actually they have done studies which show they can almost, in some cases, reverse the effects of, say, a paralysis, where people can't use their legs. So they are doing stem cell research. The stem cells, though, because they come from an embryo, an early embryo, there's people that are opposed to that, because the embryo doesn't get a choice. Embryos will be destroyed by the extraction of those stem cells, and that can obviously cause harm to the unborn child that embryo is. You can actually find stem cells in other places. They can be got from bone marrow, where people can actually volunteer to donate their stem cells, their bone marrow. But that process is where they have to actually drill in through the thigh and extract those from the bone, from the middle of the bone, and it actually can be a painful procedure. The advantages, though, are that those people who donate it get a choice, so they can actually donate the stem cells for the research, whereas an embryonic stem cells, the embryo doesn't have a choice. So recapping, the important bits about mitosis is it makes all the cells in the body, including mitose. It's used for growth and repair. It only divides once, and it produces identical daughter cells with the same number of chromosomes as the cell that started. 46 in the case of a human. And meiosis made me, so it made the sex cells, the gametes, the sperm and the egg that produces life. In that, it divides twice. Once, then twice. And it produces 4 new daughter cells, which have half the number of chromosomes of the parent cell. Okay? So that's the important bits to remember about the two processes. Now, why that's important, particularly meiosis, is because it leads to single forms of genes, single bits of DNA, which are genes, which can be combined together to make different characteristics. They're all controlled by the genes that you have, and the single form of a gene is called an allele. Now, that's a definition that can come up. They can ask you what is an allele. It's the single form of a gene, and when you combine them together, 2 alleles, you actually make a gene that will control a certain characteristic. Now, in GCSE, we often represent alleles as letters. You can use a big letter for a dominant allele. Okay, I'm going to use D as my example. So a big D is a dominant allele. And dominant means if you have any of that gene, or any of that allele, I should say, in your DNA, you will have that characteristics because it's dominant, it takes over. And small letters are used for recessive alleles, such as a little d. And there's different combinations you could have for the alleles that a certain person could have, and they would depend on what characteristics they had, what physical characteristics. Now if I use the D's as an example, you could have two big D's, two of the big dominant D alleles. And that's called a genotype. So this is the genotype of a person. They have two of these dominant alleles. You could have a big D and a little d. That would be another combination that you could have. Now, whatever the dominant characteristic would be, they would have that dominant characteristic because they've got one of the big Ds. Okay? You could also have two of the little Ds. Okay? You could have two little Ds. And because you've got two of the recessive genes or alleles, you would have whatever the recessive characteristic is. An example is tongue rolling is controlled by just one gene or one set of alleles. So tongue roll is the ability to do this. So you can try, see if you can do that. If you can, you've got the dominant gene or allele. You've got the dominant. You are either this or this because you can roll your tongue. If you cannot roll your tongue, then you are recessive. You have two of the recessive genes. This is called the genotype, as I said. This is the genotype that people have. And the phenotype is what physical characteristics they have. So this person, as I said, because it's dominant, can roll their tongue. Okay, this person, because they have one of the dominant genes, they can also roll their tongue. And this person, because they have two of the recessive, they can't roll tongue. Okay, so that's the phenotype. Either you can roll your tongue if you have any of the dominant gene, that's rolling your tongue. The only way you can't roll your tongue is if you have two of the recessive genes in your genotype or alleles in your genotype. Some other key words that you may come across are homo and heterozygous. Okay? Homo literally means the same. Okay? So homozygous means you've got two of the same alleles. So this is actually homozygous. Okay? Because they're both big Ds. This is homozygous because they're two of the little Ds. And then in the middle, this one, which has got one of each, is actually heterozygous because hetero means different. So this is heterozygous. Okay? Because it's got two different alleles in the gene, so controlling that characteristic. Okay, so homo, they're both the same, and hetero means one of each. The only combination where you can have the recessive characteristic is if you are homozygous with two of the recessive genes or alleles. And you have to, when you're talking about this, you have to refer to them and you have to write them out as big letters and small letters. And you have to say this is a big D and this is a little d for the genes or for the alleles, because otherwise it's not very clear in what you're talking about. Okay? So, with that, what can I do with that? Okay? You can do things called a genetic cross. Once you know the genotypes of the people involved, the mother and father of a child, you can work out the probability of their child having a certain characteristic, having a certain phenotype. Okay? And it's called a genetic cross. This thing is called like a Punnett square. It's the easiest way to do it. It's actually the only way I'm going to show to do it today, because I think it's the easiest by far. And you can do it for gender. So this is to determine the gender. It shows the probability of a child being born with a certain phenotype. In this case, we're talking male or female. And the probability can be shown in a number of ways. Now, Gender is only controlled by one pair of chromosomes. So one pair, one set of chromosomes control which gender a person is. If you are female, you have the chromosomes XX. You have two XX chromosomes. Whereas if you're male, you actually have an XY chromosome. Okay? So every genetic cross diagram for determining gender must be this cross, because it's the only way, because it has to be a male and a female to reproduce. So the female with two X's in your Punnett square, you put the combinations at the top. So you draw them out at the top, the two genes or two alleles, or in this case chromosomes, for the mother. And then for the father, you do the same on the side. It doesn't actually matter if you put the father up here and the mother down there. You can do that either way round. Okay? The important bit then is to do the cross that you get to give these different combinations of offspring or children. You take the letter from here and the letter from here, X and X, and that gives you the combination for that child. You take the letter from here, the letter from here, X and X, to get the combination for this child. Similarly here and here, here and here. Okay? So you end up with 4 different outcomes in your Punnett square. Now remember, I said if you're XX, you are female. So these two are female. These two children are female. And these two are male, XY, because that's the combination for male. This means if these two people were to have a child, the probability that their child will be female or male is 50-50 or 50% or 1-to-1 ratio. Okay? For gender, it's always that. It's always 50%. Okay? It's always 50% because this is always the combination that will happen. Okay? That does not mean if these people have 4 children, they will have 2 boys and 2 girls guaranteed. It means every time they have a child, they will actually have a 50-50 chance of having a girl or a boy. And we know that, okay? That's going to be the case, okay? Has to be, really, to keep the balance of genders on the planet. So it's 50-50. You can say that as 50%, you can say it as 1-to-1 ratio, you could say like 2 in 4 or 1 in 2 chance. You could say it in a number of different ways. That's for gender. You can do different genetic crosses for characteristics like tongue rolling. So we'll have a go at that. Okay? If we're talking about tongue rolling, we can pick any of these parents. Now I could be given the genotype of the parents, or I could be asked to work it out. I would know automatically if they have the recessive phenotype that they must be 2 of the small letters. I would already know that. And then it would be the person who had the dominant characteristic that I wouldn't know whether they were homozygous or heterozygous unless I had some more information. Let's assume that I've got one parent, doesn't matter, mother or father, who can roll their tongue and they are heterozygous and have this combination genotype. And I have another parent who is homozygous with the recessive, so cannot roll their tongue. To do my genetic cross, I would draw out my grid. Okay? And this is where I'd put my combinations. I would do whichever parent at the top, so I'm going to do the parent who cannot roll their tongue, so who I know must have the combination of 2 of the recessive genes or alleles. And then I would do the parent here who can roll their tongue, but I've been told they are heterozygous. This person can also be referred to as a carrier because they have the dominant characteristic, but they carry the gene for the recessive. And that can be important because sometimes they talk about genetic disorders such as cystic fibrosis. You could not have cystic fibrosis because to not have it is dominant, but you could carry the allele for it, and in which case you'd be a carrier. So this person is a carrier for not being able to roll your tongue. So then I do my cross diagram where I go across and I look here and I've got a big D, so I put that there, and I've got a little d which goes in here. Okay? And then I've got a big D which carries on across here, and a little d which goes here. Then I've got a little d and a little d to make up that combination, and little d and little d. Now I look at the children that are born from that cross. I've got two which have the dominant gene, so they will have the dominant characteristic. These two can roll their tongue. And I have two that cannot roll their tongue, have two of the recessive characteristic, so they cannot roll their tongue. So the chances of having a child who could not roll their tongue from this genetic cross, I would say, is 50%. Okay? That's the way I would do it, as a percentage. Okay? So that would be that genetic cross. Now, if you have anyone who's homozygous with the dominant characteristic, all of their children will have the dominant characteristic or the dominant phenotype. Because if you do any of that cross, which I'll do quickly here, it will always end up with the big D. So we'll do our little Punnett square again. And you can write these out in exam questions, that's fine. I will do the two of the dominant here, and then I will do, well, let's say with someone who is recessive, okay, has the recessive characteristic, the dominant is always going to take over from that. So all of these combinations will have the dominant gene in them. Okay? If I try it, all of them are going to have the dominant gene. So all of them will have the dominant characteristic. The chances of not being able to roll your tongue, chance of no tongue rolling, unfortunately for these people, is going to be 0%, because all of the combinations have the dominant characteristic or the dominant gene which is in there. Okay? And in the question you need to say the actual percentage at the end, because that's what they're looking for, that's what they're actually looking for in the question, that actual percentage or ratio or fraction if you want to express it like that. But I would say stick to percentage personally. They like to ask questions about this in a like what's called a pedigree analysis, which is a family tree to you and me. It's one of the harder things and it's a higher level skill, but if it did come up, what I would suggest, the top tips I can give you, is identify the genotypes of the recessive people. Because people who have the recessive characteristic, which in this case is sickle cell anemia, have to have two of the recessive genes. You can write that by them, and I would suggest you write it on your family tree. They must have a small d, or whatever letter they're using. This must be small, 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 small. And I can do that right away. Okay, so I can do that early on. Then, if someone has a homozygous dominant alleles, then none of their children will have the recessive characteristic. So if any of their children have the recessive characteristic, they must be carriers, even if they don't have that, that gene, if they don't have that characteristic themselves, that phenotype. They must be carriers, because if they were both dominant, okay, all of their children would have the dominant characteristic. What I mean by that is, this is Alfie here, this is Esme. She's, I know, two of the recessive. Him, I don't know, because he could be a carrier, or he could have two of the dominant. But if I look at his children, some of his children have sickle cell anemia, have the recessive disorder. So he must not be like homozygous with the dominant. Otherwise, none of his children would have it. So he must be a carrier. He must be this, big D, little d, because some of his children do. Now, it doesn't mean, again, that if it's a 50-50 chance, if you have 4 children, 2 will have it, 2 won't, it always is just a percentage chance or a probability. So you can't guarantee it. It just means every time you have a child, there will be that chance every time you have a child. So in this, we know the genotypes of these children as well. They must be 2 of the recessive genes. Thomas, we can safely assume, because this is 2 of the little d's, that he is a carrier as well. Theo, don't know what his parents were like. But again, comes from a different family, so we don't actually know again, because we can look down here and we can see that we know what Abby is, and we know that she's had a child with someone who doesn't have it, that has sickle cell anemia, has the recessive characteristic. So Theo must be a carrier as well, because if he was 2 of the dominant, all of his children would have none of the recessive characteristic, but he's already got a child that has that recessive characteristic. So he must be a carrier. So last of all, if we do that genetic cross for someone who is has the recessive characteristic with someone who is a carrier but doesn't have it, again, if we do this cross, we've got a big D, little d, a big D and a little d, a little d and a little d, and a little d and a little d, similar to the first one I did. 50% chance that Bob here will have sickle cell anemia. We can't tell which way it's going to go, but it's a 50-50 chance either way. And every time they have a child, it will be a 50-50 chance. Okay, I think I'm pretty much at the end. Okay, I'm just going to go a little bit over because I'm going to go quickly into just one more thing that I wanted to cover, because it's the only real thing I haven't covered in any detail yet, which is speciation, and it's just about what members of the species can happen to members of a species if they become separated, or isolated is what we call it. Isolation is where something separates two members of the same species. So maybe a mountain range forms on an island, or a river on an island, and the animals that used to be the same species get separated by something. They can evolve differently if the environments they live in are different. I actually show this video, which is in some people called the Lumpties, and one of them are in a warm environment where all the food is high up, and one of them is in a very cold environment, and they evolve differently because they become separated by the land spreading apart. It happens through natural selection, which is actually in core, but you still need to be able to talk about natural selection. Mutations in the DNA lead to variation. Some characteristics are more successful than others. Those people who have the successful characteristics live longer, survive longer, and are able to reproduce, and then they pass on that characteristic to their offspring. Eventually, we know it's become a new species because the definition of a new species is when they can no longer breed with members of the old species. So that's when it's become a new species, when the definition of a species is when it cannot interbreed and produce fertile offspring with members of a different species. Okay? And how we can tell things evolved is we look at fossil records. What a fossil is the remains of an organism preserved in rock for millions of years, and we can use it to find out evidence of how things evolved, and also maybe how things go extinct. Ways that those animals could go extinct, or plants, could be there's a new predator, but you have to say a new predator, you can't just say a predator. A new disease appears that wipes them out. Could be due to the loss of their habitat, due to a natural disaster such as an earthquake or volcano or tsunami, or a more successful competitor arrives that actually takes over, such as the grey squirrel taking over from our red squirrel and pretty much wiping it out because it's a more successful competitor. Okay. So we've gone through a lot, we've gone through everything in B2. Some of the things that I've covered in a bit of detail, some things I've gone over quite quickly, such as I didn't go too much into specialised cells, but I went into inheritance a little bit of detail. I went into digestion in a bit of detail. I went into the structure of the leaf in a bit more detail. Okay, make sure you do lots of revision tonight, and I'll be in at 7 o'clock tomorrow, so nice and early, for those who want to turn up early before the exam, which starts at 9. I'll either be in the world famous 104, or maybe in 106, depending on when I get there and who's here. But otherwise, if you want to come in early tomorrow, please do. And if I don't see you, just best of luck with your exam tomorrow. Hope it goes well. Thanks for watching.